Hey everyone, and welcome back. We're going to talk about the viral cycle in animal hosts. So there are basically 5 steps to animal virus infections. The first is attachment, which we'll talk about first, which is why it's in red. Then we have entry via either endocytosis or membrane fusion. And uncoding is part of that second step. So you're uncoding that DNA or the RNA. Biosynthesis, which is the most complex step and most variable depending on the type of nucleic acid. And then we have assembly and release. So this figure here came from my microbiology in OpenStax. It has all 6 or all the steps together. So you can look at that as well. But I'm going to go through each step individually right now. If I can get this to move. Excellent. All right, so the first step is the attachment. So we're going to attach from the original or from the virus to the host cell. The virus has receptors that have to match the host cell. What this does is it limits the virus's infectability. Viruses can only infect certain types of cells. Most viruses are very specific to species, which is why dogs don't contract certain human diseases. And but it's not always true. So I should say but that's not always true. The example, rabies is a virus that infects lots of different animals. The next step is the entry via either endocytosis or membrane fusion. So the cell is going to, the host cell is going to either envelop the entire structure and pull it in. That would be endocytosis. Or the membrane of the enveloped virus binds to the membrane of the plasma membrane of the cell, and the cell takes in the structure. And then the uncoding is the release of the DNA or RNA. So here you can see the penetration and uncoding. Here's the attachment. Here's membrane fusion. So the cell is bound, and now you have the nucleocapsid being released into the cytoplasm. Here's uncoding. So all we have left is the capsid with the nucleic acid coming out now. In endocytosis, the cell envelops the entire virus, pulls it in, and then that vesicle releases the virus and the DNA in the uncoding step. So the next step is biosynthesis. This is the one that we're going to spend the majority of our time on. Biosynthesis is the replication of new viral particles, but this depends on the number of, or the type of, the type of nucleic acid that's available. And so you can have DNA viruses that are single-stranded, so SS, or double-stranded. You can also have RNA viruses that can be single-stranded or double-stranded, okay? And then we have some viruses that include reverse transcribing. So these are retroviruses. So let's look at DNA viruses first. So we all know what DNA is. DNA has a coding and a non-coding strand. So if you have a double-stranded DNA virus, that's really easy because it's just like any other DNA. The minus strand is what we use as our template. And we produce messenger RNA from it. The plus strand can be used to make more minus strands. If it's single-stranded, DNA, then we're going to use the minus strand and transcribe that messenger RNA. So if we have single-stranded DNA, we can produce the complement so that we have more single-stranded DNA structures that all can be used to produce new viral particles. So if it's the plus strand of the single-stranded DNA, say it's a plus, then we make the complement first, and then that complement is the minus strand, and we can make more copies of the viral particles through transcription and then translation. RNA viruses are a little bit more complex. So most RNA viruses are single-stranded, but that doesn't mean they all are. So we have two different types of RNA viruses that are single-stranded, though. We have plus-stranded RNA viruses and minus-stranded RNA viruses, okay? A plus-stranded RNA virus is the messenger RNA that we care about. So we can directly translate it. But the minus strand would first have to be used as a template to produce the plus strand. And if we had double-stranded RNA virus, then we can use the plus strand to produce messenger RNA, and the minus strand can be used to produce more of the plus strand. So here's a plus-stranded messenger RNA that is going to directly synthesize the protein for the virus right here. And we can also synthesize the minus strand of make complements of this, which would be the minus strand, which then can be used to synthesize more plus-stranded RNA. So if we have minus-stranded RNA, we have to utilize the minus strand, use RNA, viral RNA and replicase to produce the positive strand, because the positive strand is the messenger RNA that we care about. So the messenger, or the negative RNA strand, is going to be used to synthesize a positive strand. The positive strand then can be used to produce the protein, and then we also can use a positive strand to produce new complementary minus-stranded RNA, which then can be used as a viral genome. Double-stranded RNA then has both the positive and the negative strand. So the positive strand can be used for translation. The negative strand can be used to produce more positive strand, and then we have that double-stranded RNA, again, for the genome. So we have these different viruses, both DNA and RNA viruses. DNA viruses, so all viruses can mutate and do mutate, but RNA viruses don't have the DNA polymerase to proofread the information, and therefore RNA viruses tend to mutate much more rapidly than DNA viruses. And this can actually lead to a process known as antigenic variation or antigenic drift. Antigenic drift occurs when the structure of the antigen or the surface proteins changes slightly. So in an antigenic drift, your immune system recognizes the antigen. So say this is the antigen right here, okay? So we have this green square and this red ball right on top. So say this is an antigen, our immune system recognizes this antigen. In antigenic drift, something small changes. So I don't know if you noticed, but this one doesn't have a very dark outline, neither does the box, but here they both have a dark outline. So there's been a change. This is an antigenic drift. The immune system will eventually be able to recognize this, but it will take a little bit of time. So it'd be like having this with a line on it. So it's the same structure, but now there's something different. The immune system is going to look at it and say, wait, is that the same thing or is it not? And so antigenic drift can cause problems with our immune system. Our immune system doesn't recognize, and depending on how often the antigenic drift occurs, it can actually lead to a lot of problems. But in RNA viruses, typically the antigenic drift isn't going to cause severe epidemics or pandemics. What does happen that, whoa. Okay, hold on a second. I have to fix that. That was weird, wasn't it? So another phenomenon known as antigenic shift can occur. And when that occurs, this can actually lead to pandemics, epidemics, serious outbreaks. So in this case, the immune system recognizes, again, this pathogen here, this is the virus, but if it changes completely, where now it looks very different, then that's known as antigenic shift, and the immune system will not recognize this at all. So this is a brand new pathogen the immune system doesn't recognize and can't produce any protection against. It would have to start from scratch on this one. And then we have special viruses, which are called retroviruses. Retroviruses have single-stranded positive strand RNA, but they also contain an enzyme known as reverse transcriptase. Reverse transcriptase can synthesize DNA from your RNA. And so here we have this positive stranded RNA right here. Reverse transcriptase is used to produce a single-stranded negative strand DNA. Well, then DNA polymerase in the cell can produce the positive DNA strand, and this can then enter into the host genome. So it can incorporate itself in the host genome and become a provirus. Of course, you also can use this positive stranded RNA to synthesize new proteins, viral proteins, that are then able to produce more virus, which then can infect more cells. And so this is what HIV does. So let's go through the HIV cycle. HIV binds to a CD4 cell, so this is a helper T cell receptor, and then it enters. So it moves in, and once it's inside, the viral RNA is going to be reverse transcribed into negative stranded DNA, which then can enter into the nucleus. Inside the nucleus, we can then produce, using DNA polymerase, the positive stranded DNA, and this can get incorporated into the cell. So now we have a provirus. This provirus now can produce more positive stranded RNA virus structures that can infect new cells, more T cells. And so this is similar to the lysogenic cycle in bacteria. Why can't I change this? There we go. Sorry. The last two steps then are assembly and release. So assembly and maturation are just the production of the new fully complete virus. And then release occurs when the virus leaves the cell. So viruses, when they leave the cell, will leave via budding or via apoptosis, depending on the type of cell or the type of virus and the type of process that occurs. So if it's an enveloped virus, typically they release via budding, whereas non-enveloped viruses will lysis open the cell. So the cell basically bursts and the cell dies. And so here you can see that budding process where the enveloped virus just gets covered in the plasma membrane and then releases via that budding process. Apoptosis then would be where the cell just bursts and the virus pops out. I'm going to stop here and we'll get into the different types of infections in our next video.